Grade 12s, welcome back, and gosh, are we excited for a good lesson.
What we've got here is a protein synthesis structure. What I wanted to show you here is that you have your nucleus. This is your nuclear membrane, and I'm going to give it a little bit of a color here. There's your nuclear membrane, and in between the nuclear membrane you have a pore. And this is where the little messenger RNA is going to move out. Now remember, your takeaway joint is your ribosome. Okay, that's where the hamburger is going to get made. Or in this case, the amino acids are going to be put together to form pieces of proteins. Remember, your amino acids group together to form polypeptides, and the polypeptides form proteins. Proteins are huge molecules. So we're now making little amino acids at a time. So we're saying, right, now, how are we going to do this? The information comes from our DNA. So here we have our DNA strand. And it's this DNA strand that now gives us the information. It provides the template. In other words, the information. The messenger RNA comes along and it codes on. Now, codes on is what makes it, and this is why we call it a codon. And that's why I prefer codon to a triplet base. All right, so it codes on. This messenger RNA codes on. It takes the information from the DNA, okay, as a complementary base set, and that's what we call transcription. I just want to get myself a
doing that smart board.
A blank screen. All right, now watch here. Okay, if my DNA strand, and please, if you understand this, you're A for away. The DNA strand, okay, remember they're going to open. So your hydrogen bonds are going to break. An enzyme causes that to happen. And those hydrogen bonds go ploops. Now the information on the DNA strand is there to get. All right, so there's the DNA strand. So now we're going to make a DNA strand. So let's do that, okay? And remember, you're going to have your phosphate backbone and your pentose sugar, and attached to your pentose sugar, which is your deoxyribose sugar, you are going to find a set of letters. Now, between A, T, G, and C, you tell me what I'm going to put here.
Well, anything.
A, T, G, or C?
T.
T.
G.
And G.
And another G. And then an A.
Okay.
Tiger.
All right. So, Indy's given us our letters. This would be what is on that DNA strand. All right. Along comes RNA. And here's my RNA. Which RNA? Our messenger RNA. And messenger RNA goes whoops into the nucleus. It goes to the DNA and it says, okay, hold on. And it does this. So I'm going to have, what is the complementary base to T, people? Remember, A to T and G to C. So if there's a T on the DNA strand, what is the complementary base? It is going to be A. And the complementary base for G would be C. And for G, you know what, I'm going to swap this around here, Indy, because I actually want to show the learners a specific thing. Okay. So let's make this A and make that one there G. All right. So what we're going to have is our messenger RNA is going to come and it's going to say complementary to T, complementary to G, and complementary to A is not thymine. It's going to be uracil if it is RNA. So uracil will clock in here. Okay, this has 3 bases. All right, it's got 3 little bases. 3 means it is a triplet base, or we say that it codes on, so it becomes a codon. This little group here of 3 is a type of messenger RNA, and it's called a codon. The codon gets its information from the DNA template. All right? Now that's going to tell it to make a specific amino acid. So it says, all right, got it. Now, the messenger RNA scoots out of here, and off it goes through the nuclear plasm and the nuclear pore. All right? And I have forgotten we are ACU. And here comes ACU. Right? So it's literally come from there and it slips out so that we have our ACU there. This is my messenger RNA. Now where does it go to? It goes to our takeaway place, which is going to be the ribosome. So it gets to the ribosome, okay, and we now have the opposite to what we had on the DNA. Okay, you understand that? It's the complementary base on the DNA. It goes to the ribosome, and when it gets to the ribosome, UCA, all right, and everybody's very, very happy here. We're going to have on this side transfer RNA is going to come along and say, all right, let's match up here. And what does it do? It is the anticodon of what was on the DNA.
Yay, anticodons!
Yes. So your codon, people, was the messenger RNA taking a sort of almost a picture of the opposite or complementary base of the DNA template.
You know what,
Okay,
Kathy?
that's your codon. The opposite is going to be your transfer RNA, which is the anticodon. It is the opposite of the messenger RNA. Okay, people, remember, I'm trying to think of a different analogy to use here, but your messenger RNA, when it goes to or at the DNA, and it makes this copy of the opposite process of what is on the DNA, we say that it is a codon. All right, so your messenger RNA codes on the little piece of 3, the information that you're going to find on the DNA. All right, this process, and I'm going to use a different color here, is called transcription. And you must know what transcription is. So transcription is when the messenger RNA comes along, groups together as 3 bases that are complementary, the codon. This little thing is a codon. And it then is the opposite or complementary base to the information that is on the DNA strand, which is the template. Now this, forming the opposite information, the codon, of the DNA information is called transcription. Okay, then we move down, and the little messenger RNA goes bitty, 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 bitty out through the nuclear pore, and there's my messenger RNA code on. All right, it's a codon because it's a little group of 3 very specific messenger RNAs that have been put together, or little RNAs that have been put together. They're very specific. It has coded onto what is in the DNA, and it is the opposite. It's the complementary base. That's transcription. Now, the messenger RNA codon, which is this little specific group of 3, comes to the ribosome. And at the ribosome, it says, okay, here I am. That's the message I have from the DNA. But the transfer RNA says, wait, I have to do the opposite to what you've got here. Why? Because it needs to look like the DNA. So it says, okay, I'm going to do the opposite. If something is anti, it is opposite. Okay? If you like chocolate cake and I am anti-chocolate cake, it means I don't like it. Which is a lie, I love it. But did you know that? So anything anti. So if the codon is ACU, what is the anticodon going to be? The anticodon is going to be the opposite to what I have on the codon, the messenger RNA codon, which is going to be, what's the opposite to A? And if you can't remember, A equals T or U, and G equals C. Okay, so how do we look at it? We say, right, A to T, it can't be T because, hello, it's RNA, so it must be U. And C opposite is G, and the complementary base to uracil is going to be adenosine. All right, now what do we have? UGA. If we go back and look at our TGA. So remember, RNA will always have a U instead of a T. So there we had the anticodon on the transfer RNA, okay, was UGA. Is it the same as the DNA? Yes. Remember that on the DNA we're going to have thymine, and on the RNA we have uracil. An easy way to do it is this, people. Let me just get I want a blank screen. Okay, look here. This is what you do. Piece of cake. Let's get my white. Let me say, right. I'm going to have 3 columns. Okay? And here we have the DNA, which is going to be the template. So that's the information on the DNA strand. There's going to be one strand of, or let's put here, one strand of DNA. So this is either the left strand, the right strand, the first strand, the second strand, whichever. Okay? Here we are going to have the messenger RNA. And this is going to be the code on, or it's going to produce the code on. And remember, the codon is the little group of 3 bases that are part of the messenger RNA. Then, in pink, all right, nice contrast, we're going to have the, oh, it's not working, the transfer RNA. And please take note, messenger and transfer is written in little letters. All right, they're lowercase, okay? And your transfer RNA is the anticodon. It is the opposite to codon. Now watch here, okay? The DNA, what did we have? We had TGA, huh? So we've got, there's my DNA strand, and a portion of this DNA strand, we are going to have TGA. Okay? TGA, the messenger RNA comes along, it forms the anticodon, and it says, okay, just those 3 little pieces. Let's just make this a little bit shorter. Okay, wait, let's just do this again. It only works with those 3 because it is a codon or triplet base. Okay, codon or triplet base. It's this little piece of RNA. And it comes along and says, right, complementary of T would be A. Complementary of G is always C. Complementary of A is thymine, but it's RNA, so therefore, what are we going to have? Uracil. Okay, that's my codon. Anticodon is opposite. So what am I going to have? I'm going to have here my transfer RNA is going to have, instead of A, it's going to have T. Instead of C, it's going to have G. And instead of U, for the U, it's going to have A. But, sorry. It's going to have a U. I'll just get my white here, fix that line up. All right, watch here now. Okay, you're going to have to just squish it in here. I'm going to use the yellow. Watch here. The DNA and the codon of the messenger RNA is opposite. So the codon is the opposite to what the information is on the DNA strand. It's opposite. And the anticodon and the codon, so the difference in the messenger RNA and the transfer RNA, it is opposite. But now, in being opposite, you've had opposite twice, which makes what is on the anticodon the same as what is on the DNA, except that because this is RNA, it is going to have uracil instead of the thymine. But the information is identical. So if you get any question where they say to you, here are the different amino acids, and this is how they blah, 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 you say, okay, fine, not a problem. If they give you the amino acid sequence will be your anticodon. So you immediately know it's from your transfer RNA, it's an anticodon. It will be the same as your DNA, except you'll have uracil there and thymine if it's DNA. That's how you know if it's DNA or RNA. DNA has thymine, RNA has uracil. And your codon and your anticodon are opposite, and your codon or your messenger RNA codon and your DNA are opposite. And if you can remember that, then you're A for away. Because what happens now is that this transfer RNA, the tRNA, of UGA, this is this little thing here. This is the anticodon, and the anticodon is always carried by the the transfer RNA. That anticodon, somewhere there is an amino acid that is going to be UGA. All right? It then picks up that UGA and it brings that UGA, which is your anticodon here, to the ribosome where the little amino acid is out through the ribosome. All the little amino acids are gathered together and they then end up forming a protein. All right, you have all your little peptide bonds, and we have this huge protein that develops through polymerization. Okay, a polymer means lots of parts to a big molecule. Rization, to make, polymerization. Okay, now, I just want to go back here to, okay, watch here. And I don't think many of you will see this. Okay? And this is so clever. It's like coding. What you've got is your first letter of the codon or triplet base, whatever the triplet base is. The three little bases, you've got the first letter of the base, the second letter of the base across the top, and down the side here, you've got the rest, which is your third letter. All right, so watch the combinations here. My first letter is either U or C or A or G. My second letter can only ever be U or C or A or G. All right? And my third letter is either UCAG or UCAG or UCAG for each of them. Now, the words you see written here are all the amino acids. There are only 20 known amino acids. Us wonderful, intelligent human beings have only ever found 20 different amino acids known to man. So there are only 20 combinations needed of your 4 nitrogenous bases here. Your 4 nitrogenous bases is the second part, because remember, instead of saying letter, the letter is actually the code. So whether it is the codon that you are going to find the opposite of this for your messenger RNA, or the actual anticodon, which is your transfer RNA information here, because what's on that transfer RNA, that anticodon, is what makes the amino acid. So have a look here. If we have first letter is U, second one is U, and the first one here is U, so I've got 3 U's in a row, I'm going to have, or UUC, that combination, I'm going to have phenylalanine. A nice one that they like is leucine. It's one that they can sometimes ask. They love valine, they love alanine. It doesn't mean you must go and sit and learn these things off by heart. They'll give you a table and say valine is your amino acid that's going to be made, all right? And your valine is made up of the following triplet bases. It's made up of GUU or GUC or GUA. They'll pick one of them, and they say GUU. Then you know what was on the DNA template. If it was GUU, it is going to be GTT. Okay, for valine. Your anticodon, your codon, which is carried by your your messenger RNA. Okay, that codon's carried by your messenger RNA is going to be the opposite, people, and the opposite to G is going to be C, the opposite to T is A, and the opposite to T is A. And then we end up going to the anticodon, which is carried by the transfer RNA, all right? And what am I going to have? I'm going to have the opposite to the codon, which is going to be, opposite to C is G. Opposite to A, hold on, remember it's RNA, so there can't be thymine, it's got to be uracil, uracil. Now look here, GUU, there we go, and you will pick that it's valine. And it's as simple as that. There is nothing complicated as long as, where did I do my table?
You know what? While you look for your table, I know you can listen and look. Neviron Felix has something great. I don't know what you think of this. If this helps anyone, he says, it's easy to remember that RNA does not drink tea. That's how I remember that there is no tea in RNA.
Thymine and uracil, perfect, beautiful. Okay.
Neviron.
Oh, there we go.
It's fantastic.
So RNA doesn't drink tea, it drinks uracil. And that's the best way to remember it. People, if you see any combination, any triplet bases, in other words, 3 little bases together, all right, all you have to do to determine if it's RNA or DNA is you turn around and you say, okay, is there a uracil? There's a uracil or a thymine. The thymine means it's DNA, and the uracil means it's RNA. Okay, that's number one. Number two, we've got two very important definitions that you must know, or terms you must know. One is transcription, and one is translation. Now, to scribe, which is where transcription comes from, to scribe means to write. Trans means to move. So you're basically moving the writing. That's what transcription is. Or you're copying it. You're transcribing. You are copying it. Okay, but the opposite. So that's transcription. Transcription is when your messenger RNA codon, all right, forms complementary bases and codes to the template of the DNA. All right, so transcription is the process whereby, sounds very complicated, eh? But transcription is the process whereby your messenger RNA codes on to the template of the DNA. And a codon has 3 triplet bases. All right, so now we've coded on, and it is the complementary bases. Okay, they are opposite. They are the complementary bases on the code on to the DNA template. So we now have the opposite. Okay, fancy word for opposite, saying it is the complementary base set. All right, now my umbilical cord that I'm moving along. Okay, we look at the anticodon. So now the messenger RNA goes zhoops, and it goes all the way to the RNA, because that's where protein synthesis is. Okay, the grade 10s learned today that the ribosome is the site for protein synthesis. This is why. Okay, now the anticodon is opposite. It is anti. It is opposite to the codon. It is the complementary to what is on the codon. So instead of A, complementary would be U, because remember RNA doesn't drink T. C to G, U to A. And what do we have here? We've got the anticodon for the amino acid. And the information, when it is anti-coded, and it's coded like that, and we end up with the amino acid, we call the reading of the transfer RNA of the mRNA, creating the complementary bases, we say that that is translation. So your transfer RNA is translating the information on the codon, right, but as an opposite complementary base. So we have transcription, the messenger RNA forms complementary bases to the information on the DNA, okay, and anticodon is when that message is translated and formed into an amino acid. So at the end of the day, what are we translating? We're translating the information from the DNA strand to make here, to make an amino acid. Amino acid passes out, and what do we end up with? A whole bunch of amino acids, peptide bonds, and we have a protein. People, that's protein synthesis in a nutshell. Adenosine to thymine, okay, or uracil in the case of RNA, and guanine to cytosine. Always. And if it has a U in it, so a nitrogenous base that's uracil, you know it's RNA. And your transfer messenger RNA is opposite. It's the anticodon is your transfer RNA. So messenger RNA codes on 3 little triplet bases. Your anticodon is your transfer RNA. And that is then translated into a specific amino acid. And if you remember this, 100% in any question you get on protein synthesis, and I think it's going to be your essay at the end of the year. There you go.
Thanks, guys.
Cheers, bye.